name is Carol Knevelman, and I'm the VP and Head of Process Development within Oxford Biomedica. So I'm responsible for developing our internal programs and also that of our collaborators. I believe that would have to be the clear demonstration of successful long-term treatment of conditions which were previously unsuccessful. I'm thinking particularly of treatments for leukemia using CAR-T therapies and stem cell therapies for ADA-SCID, for example. In CAR-T therapies, genetic modification of patients' T cells to express artificial T cell receptors in order to target particular antigens on cancer cells has led to dramatic response rates in the number of successful clinical studies. The platform was designed so the vector can be modified for delivery of genetic information to an extremely wide range of cells, including selecting targeting for gene and cell therapies. The vector has been adapted for CAR-T cancer therapy, amongst many others. One of the main challenges is providing sufficient product to the right quality. Current technologies have not caught up with this development, and as gene therapies expand from ultra-rare and rare to larger indications, there's increasing demand for greater quantities and higher quality of vector. This drives the need for further innovation in vector production platforms and vector purification in particular. Demand will also increase due to patient numbers and the amount of vector required per patient. Example, to address the lung and liver. There is therefore an opportunity for vendors to develop easily scalable and available systems from clinical through to commercial supply. To meet the forecast of vector demand for gene and cell therapies, I envisage a transition to process intensification through integrated continuous processing, automation and digitalization for data management, and single-use systems in order to aid speed to market. These provide opportunities for achieving cost-efficient large-scale vector production and achieving the right quality of vector to meet those demands. I also see a transitioning to the use of packaging cell lines in order to provide us with higher quantities of vector to the quality that is required as well. And also I see automation in analytics providing further increase in the speed of manufacture in order to increase or reduce the time to batch release.